by training, I'm an epilepsy surgeon, and I primarily work with patients that have refractory epilepsy and trying to find ways, surgical ways of treating that. I'm a neurosurgeon by training. And as part of, but I'm a surgeon scientist. So, I also go to the lab and I work on mammalian systems, mostly mice, but in general, mammalian systems to try to understand how the brain works. and how this came about is the work we do in the lab is really, it's very simple. It's it's a window into the brain. And that's pretty much what it is we remove part of the skull, and using optical microscopy, we can try to understand cell-cell interaction. So how does a neuron interact with an astrocyte and microglia? And, so the first thing I really want to understand is, what are those interactions in a live animal, first of all? And then the next step would be trying to understand disease processes. So the cell death is in almost anything from development into aging. And so I thought it was a good way to start, like what happens when a cell dies in the brain. And so that's how it started. It wasn't really built up from something, but it opens a lot of doors. So the first thing that we did is how can you kill a single cell in the brain and watch what happens to it. So rather than hitting the brain with a hammer and causing this massive amount of cell death, can you see just what happens when one single cell dies, and what are the neighboring cells going to do about it? And so to do that, we actually developed a technique. So there's a paper that actually goes with this paper that's already published. It's called two-fatal. And it's a way where we can bleach the nucleus of any cell we choose. And here we choose neurons. And then just that single cell. So you can see the cell before, during, and after. And then you can watch what happens around it. So first of all, we developed a technique called two-fatal to spatiotemporally understand the process of cell death. And then after we did that, then we said, okay, well, how are these dead cells removed? Because cell death continuously occurs during development, in some populations, just throughout life, and becomes, a lot more during, disease like a brain injury or neurodegeneration. It's just a bigger cell death process and way more complex. So just starting with one cell, and so we decided to kill one cell, and then we had to label all the other cells with different colors so we could differentiate them. And so the neurons were labeled, and the astrocytes were labeled, and the microglia was labeled. And the first thing we noticed is that, contrary to what people have thought in the past, that actually things don't occur as clearly as, people think. Microglia, remove cells and remove dead cells from the brain, but it was more, there's like a little bit more division of labor in the brain. It wasn't so clear cut that microglia was the primary, but microglia and astrocytes, they work in concert, and they both remove different parts of the cell.